So, welcome everyone. This is the Reclaim Community Meeting, our May edition. Just want to give a few quick updates before we move on to our speaker of today. So, I wanted to thank the folks who helped me with the MIT Solve application last month, and a little update on that. In that, unfortunately, we had a few last-minute complications, so we couldn't quite get that submitted in time. But I wanted to just acknowledge how much hard work you guys have helped with that application and giving us some good ideas and suggestions. So we're definitely going to consolidating all this for the next time the MIT Solve grant comes around. You've also noticed that, you may have noticed that we've had to drop the open working sessions recently, the fortnightly sessions where we just gather, drop in, drop out and work on various activities like documentation and most recently the MIT Solve. But we will be picking it up again. But in the meantime, if anyone wants to just hold a session or bring something up to discuss, feel free to let me know, and I can make sure we run those sessions and facilitate you in doing so. But for the main reason why you guys are here today, I want to introduce you to Ariel, who is a postdoc researcher. He was a postdoc researcher at the Pontificia Universidad Católica de Chile. Hopefully I pronounced that more or less okay. But now he's at another university. I didn't quite catch the name, I'm afraid, Ariel, so I might pass that to you in a sec.
Universidad.
His work was in plant synthetic biology, and he did a little switch during the pandemic to work on things a bit more like this, the SARS-CoV-2 detection. But he has also had experience in doing DNA assembly methods, Golden Gate loop assembly in plants, universal loop assembly for potentially any other organism. And I think he gets quite stuck in a lot of molecular biology, educational tools and workshops and stuff like this. But for today, he'll be telling us more about their recent publication. And we have a bit more time, we can hear about all the additional work you've done outside of this. So without much further ado, I'll pass you on to Ariel.
Okay. Do you hear me well? Because I have noise in my house, so tell me if you don't hear me.
All good. You could go.
Well, hello everyone. First of all, thank you very much for allowing us the opportunity to share our experience with this project. Well, my name is Ariel Cerda. Today I will briefly discuss the significance of our work titled An Open One-Step RT-qPCR for SARS-CoV detection, which was published in PLOS ONE early this year. And this project is important because this project is a collaborative effort involving many individuals, primarily from the Millennium Institute of Integrative Biology, which is part of Universidad Católica, and also part of this collaborative effort known as Reclaim. Well, the context leading to this collaboration is clear, as the COVID-19 pandemic has affected us all in one way or another. According to the latest epidemiological report, there are currently over 700 million cases worldwide, and more than 7 million people have died due to this disease. And additionally, in our specific case in Chile, where most of the research was conducted, the situation remains critical with one of the highest rates of confirmed cases. Faced with this challenge, from our research perspective, this collaborative effort was born, involving a large number of people who are not usually involved with clinical diagnostics, aiming to contribute open source solutions to combat the pandemic. For instance, as Jan said, at that time I was working on my doctoral thesis in plant synthetic biology, and I had never before worked on solutions related to human clinical diagnostic. As many as you know, as a result of the high number of cases, there was an overwhelming burden on the local health system, accompanied by a persistent shortage of the testing supplies and personal protective equipment. In Chile, this is severely affected early diagnosis and impeded the control of monitoring of virus transmission. Moreover, this led to a significant underestimation of the number of infected individuals, posing challenge for the efficient management of health resources. And this scarcity of resource for clinical diagnostic was clearly reflected on the headline of local media. And this was not just a local or Latin American issue, but a global one, with many media from different parts of the world reporting similar supply shortage. Considering the rapid evolution of the virus and the emergence of various strains through the crisis, well, rapid and efficient identification is crucial for implementing control measures that limit the spread of the disease. The main challenge for that include detecting small amounts of viral RNA to reduce the number of false negatives, distinguish signals between different pathogens to decrease the number of false positives, have the capacity to test a large number of patients while avoiding cross-contamination. To date, all these challenges are effectively addressed by the gold standard technique, RT-qPCR. And one of the lessons that this pandemic has taught us is that open and global collaboration greatly contribute to finding solutions. Moreover, this crisis has highlighted that there is a need of simple and cost-effective detection method to quickly identify the SARS-CoV virus, that there is a need of critical molecular supplies that can be easily and locally produced, especially in developing countries, and that the enzymes that are needed to this kind of test, as a reverse transcriptase and DNA polymerase, are subject to centralized production models and intellectual property restriction. This is where our involvement in the beginnings of Reclone comes into play, aiming to facilitate the access to reagents, protocol, low-cost methods, and other means to overcome the challenge of reagents scarcity. Initially, we decided to focus on the two steps most affected by reagents scarcity in our country: the viral RNA extraction for patient samples and the amplification detection system. To address the shortage of RNA extraction kits, nasopharyngeal samples were typically pooled, or alternative homemade extraction techniques were used. In fact, we published a straightforward paper describing a simple method for RNA extraction to detect SARS-CoV by RT-qPCR. And as for viral RNA amplification and detection system, which typically involve RT-qPCR, we decided to develop an open one-step RT-qPCR kit. This decision was mainly because one-step procedures are preferred, as they help to avoid potential pre-analytical errors that can occur during samples preparation, optimize reaction times, and facilitate automation. Additionally, we decided to work in two parallel options. One relies on a specific probes, and another based on dyes with affinity for double-strand DNA. To create an open-source master mix for probe-based one-step RT-qPCR, we express and purify specific recombinant enzymes. We use Moloni murine leukemia virus reverse transcriptase, or MMLV reverse transcriptase, for generating the cDNA. And additionally, the thermostable Taq DNA polymerase was employed, which not only catalyzes the polymerization of nucleotide in double-strand DNA, but also exhibits exonuclease activity that is required for this kind of assays. This activity is crucial as it allows for effective use of probes in our assays. And for the same assay, but independent of probes, based on a dye, we use the same reverse transcriptase, but replace the tag DNA polymerase with PFU fused to the 7D domain. This domain prevents polymerase dissociation during amplification and improving processivity. And it also facilitates amplification in samples containing PCR inhibitors and GC-rich reagents. Briefly, all enzymes were obtained through IPTG-induced protein overexpression in E. coli and purified using a two-step process. First, we use a nickel NTA immobilized metal affinity chromatography, followed by heparin affinity chromatography, leveraging the fact that all the enzymes bind DNA. And we then pull the protein containing fraction and analyze them by SDPH, where we consistently observe the expected size on the gels. Additionally, to support local enzyme production, for this study, we provide detailed purification protocol for each enzyme on the open access platform protocols IO. So if you have any question about these protocols, they are all available in these links. These links are explicit in our paper. Next, we tested the activity of the purified enzymes through two experimental approaches. Initially, PFU and MLB-RT we used in a two-step RT-qPCR assays with a known RNA sample. We experiment with different commercial reaction buffer and reducing agents, specifically beta-mercaptoethanol and DTT. In all cases, we obtained DNA bands of the expected size, but the best result came with DTT. Following this, we tested a homemade reaction buffer. In fact, it's a non-proprietary buffer in a one-step RT-PCR setup using MBM-LV-RT for cDNA synthesis and either tag DNA polymerase or PFU for DNA amplification. We consistently achieve bands of the expected size using different concentration of total RNA treated with DNA as a template. It's worth to mention that at this step of this study, we didn't have RNA for patients, so we test with plant DNA, plant RNA. So, but what's the first step to establish the buffer and the condition that allow the activity of the enzymes? Once we tested and validate the components of the one-step RT-qPCR, we evaluated the homemade master mix using synthetic SARS-CoV-2 RNA samples. We began by in vitro transcribing the SARS-CoV nucleocapsid or engine, purifying it, and then serially diluting it to achieve different target concentration. And additionally, we synthesize RNA for other engines as MERS-CoV and SARS-CoV-1 as negative controls. For the amplification of this target, we know that there is a lot of primers out there, but we use the CDC recommended primers and double quenched probes to amplify the N1 target and N2 target that have 72 base pairs and 67 BP fragments within the SARS-CoV engine. After we perform a proof-based assays using the reverse transcriptase activity of MMLV and the DNA polymerase activity of tag DNA pol, in vitro transcribed viral RNA. Here we are looking at representative amplification curves with N1 and N2 CDC approved double quench proofs. Each curve represents a specific dilution of synthetic RNA ranging from 10 to 10 million copies. And as we can see, you can see the probe-based assays successfully detects RNA copies of the SARS-CoV gene with both primer sets, identifying as few as 10 copies per reaction. This level of detection is comparable to some approved SARS-CoV-2 kits and other open RT-qPCR methods. And importantly, no signal was obtained in non-template controls or other synthetic RNAs from MERS-CoV or SARS-CoV-1. In parallel, we assessed a dye-based RT-qPCR using the MLV, but PFU as polymerase in a proof-independent alternative. This system utilizes the same primers, N1 and N2, and an intercalating dye, in this case Evergreen, providing a cost-effective alternative to Tachman probes. On the right, you can see representative results similar to those previously shown using synthetic RNA templates. We also perform a melting curve analysis to confirm a specific product amplification, as the fluorescent dyes from or the fluorescent signal from the intercalating dye is not sequence specific. The dye-based one-step RT-qPCR assays detects about 100 copies of the synthetic SARS-CoV-2, which is significant lower than the LOD of commercial kits. Moreover, we observe non-specific amplification in non-template control and low concentration RNA samples, likely due to cross-contamination or primary dimers, despite the efforts to optimize this master mix. We consistently obtain this kind of non-specific amplification. This results on the recommendation of clinical laboratory that we are working for. This suggests that a probe-based approach using the homemade MLV RT and Taq DNA polymerase is more effective for detecting synthetic SARS-CoV-2 RNA and offers a promising option for further clinical sample testing. This was the moment when we have to decide between the two steps, and by recommendation of the clinical laboratories, we decide to go further with the probe-based approach, because we have a lot of problems with this. We figured out a lot of solutions, but at the end, the clinical laboratory say just use the probe assay. And finally, after confirming that the accuracy and the sensitivity of our probe-based master mix for detecting synthetic SARS-CoV RNA, we validated using clinical samples. So nasopharyngeal swabs from potential COVID-19 patients from Chile were collected during the pandemic. These samples were analyzed and categorized in a clinical laboratory first into 20 positive and 20 negative samples, following the CDC recommended RT-qPCR guidelines. And we use the same primers that we're using, the N1 and N2 targets of the SARS-CoV-2 nucleocapsid protein. And additionally, we add and we amplify the human RNA-P gene target as an internal control. We evaluated 40 clinical nasopharyngeal samples using two kits, a commercial one and homemade Taqman probe-based one-step fraction mix. And the probe-based mix produced results comparable to the commercial kit, as shown by the similar CQ values for each primer probe set. And these assays demonstrated 100% specificity with no false positive among previously negative samples. And additionally, it confirmed nearly all previously positive samples achieving a 95% of sensitivity. Despite the non-specific amplification from the homemade dye-based mic, we collected 33 nasopharyngeal swabs from symptomatic individuals in Chile in the UC Christus Health Network that is very close to the Universidad Católica. And of these samples, 24 result positive and 9 result negative. This was determined for the clinical laboratory using the same recommended CDC primer and no probes. And we analyzed those samples using two kits, a commercial one and our own probe-based RT-qPCR. And as shown in the figures, both reactions mixes detected the virus in all positive samples. However, the homemade assays inaccurately identified 3 negative samples as positive and 6 as indeterminate, showing us insufficient specificity for clinical analysis. Despite this, there was a high correlation in CQ values for positive samples between both kits. At first, we for one of the reviewers, they say us that maybe we have to let out this assay, the dye assay, because it didn't work well. But the correlation between the positive samples say us something, so we decide to let in the paper because we think that it was very valuable information for anyone who want to go further in that kind of assay. So that's the reason because we let it in the paper. So as conclusion and discussion, well, the pandemic underscored the vital role of clinical labs in managing COVID-19 and highlighted the significant impact on open science by promoting open access, open source. These communities not only facilitated swift and broad responses, but accelerate the search and democratizing vital information of this kind of research. Our homemade probe-based one-step RT-qPCR reaction MIP has successfully detected SARS-CoV in clinical samples, delivering CQ's values on par with those of commercial kits. This highlights its potential for local production, especially in low or middle-income countries, making it a practical solution in resource-limiting settings. Well, day-based RT-qPCR assays offers an affordable alternative in resource-limited settings, but require careful handling due to the risk of non-specific amplification. We think that strategies like establishing a precise cutoff cycles or using contamination control measures can mitigate some of this challenge, making these assays viable yet demanding option for cost-sensitive environments. Our study indicates that enzymes of moderate purity are still effective for RT-qPCR assays. This is crucial for resource-limited settings where high purity levels are hard to achieve and maintain. We think that locally produced enzymes can reduce costs and improve access to essential diagnostic tools without compromising performance. And finally, thank you all, the researchers at the Millennium Institute of Integrative Biology, for making this research possible and bringing everyone involved together for a great cause. A special thanks also to those who bridged the gap with clinical diagnostic expertise, helping secure the samples needed to test our enzymes. I like to acknowledge Gina Daly, Thomas Graham, Robert Dian, and Xavier Darzac from the University of California, Berkeley, for sharing the TechDNA poll plasmid. And additionally, we want to say thanks to the members of GLAM Consortium, the Reclone Forum, the Open Bioeconomy Lab, and the Just One Giant Lab Open COVID community for the invaluable advice and for sharing essential information that aided the establishment of diagnostic reaction for SARS-CoV. That's all.
That's great. Thank you very much, Ariel, for presenting that bit of work. Shall I open the floor to the community? Do you have any questions for Ariel? Definitely quite a few. Oh, okay, Tracy, do you want to go ahead?
Yeah, I'm gonna ask a very related question to my research because I'm also using this PFU. I think this PFU in particular. What is the duration before expiry for the kit that you designed?
No. That's a very good question. The reviewers also question a lot of the storage condition or the half-life of our enzymes, and we tested our enzyme during a long period. I mean, we were doing these assays with the same batch of enzyme during 1 year, and we always obtain the same results. But I think we don't have the people to test quality controls or the the people that allow us to establish a product that assures the life of the enzymes in our test. So I can say that at least 1 year in minus 20 degrees, but it's something that we have to explore because we established the condition minimal for this assay, but we have never had do a assay that we have never tested that kind of duration of the RT. But we have experience with we use this kit in our course and we have used the same enzyme during 2 years now. So unless we have an idea of what is the shelf life, but you are right. We need to test these.
I can hopefully, if I finish my PhD anytime soon, I can provide some information because I'm using the PFU enzyme with the SSO7D also part. So
what is your experience
there are many strategies that can be applied. One of them is making cellular reagents, like Dr. Jenny suggested, and there are some alterations using some buffers. But what I found that could be helpful, and I'm now testing to see, is that using arginine itself by itself as an amino acid, some patents said that it could provide some long and durable stability for at least 2 years in a room temperature or a fridge, because you know that we don't have some countries don't have electricity to have minus 20 all the time, and cold supply chains don't have the means to have this constant minus 20. So I'll be happy to share once I get these results.
Oh, thank you. We have very good results with the stability enzyme, but using trialose in our buffers. But for our kind of setup, trialose was very expensive, so we preferred not to use it. But we know that with trialose we can let the enzymes on room temperature, and we have no problem with that.
Oh. I've read about it, but also found it expensive also. Thank you.
Thank you.
Any more questions in the room for Ariel? So is anyone following up with this work and continuing this research? Do you know?
Maybe Fernán can help me with that.
Yes.
But we still are using that enzymes on our course. But I know that actually there is nobody working on something bigger than that. But maybe Fernán can help me.
Yes. Hi. Yeah, we have been using with an Ariel already mentioned the collaborators in the project like Cesar and other people in our lab. Some students kept working with some of the enzymes for example, preparing samples for PCR fragments to be sequenced, for example. So Paola, an student from Peru in our group, is working on she's studying waterborne pathogens, Cryptosporidium. So she put together a protocol for using the PFU enzyme to reduce the cost of that work, and has worked really nicely. With the RT, the reverse transcriptases, we have used them in combination with BST enzymes for LAMP reaction, and there are other students working on the detection of other waterborne pathogens like Giardias, also for environmental DNA, and another student working with agricultural pathogens like PBY and others, new pathogens that are arising in the north of Chile, in Arica. I mention this because this is something that we always like to say, that by being able to produce all these enzymes, thanks to the collaboration with the lab of Jenny and members of the Reclone network, it opened many doors and many horizons for research in our group that otherwise is impossible for us to do, especially in the LAMP, using LAMP reactions. They are very expensive for us to buy here. So the local production and being able to work with the group of Cesar and Jenny has been super useful, and all the projects that I mentioned were created after we started producing these enzymes, and not before, because it was too expensive. And then on the second project, we use this a lot for education. We combine all these reagents with open hardware, like open hardware that is designed by our group and by other groups, like the qPCR, the GMO detective, and lyophilization method, so we can implement a practical hands-on teaching in different courses that before it was, again, prohibitively expensive due to the cost of these reagents. And I should mention that now we are trying to expand the use of these resources in other universities, for example, collaborating with people in Argentina to create this kind of like open educational resources, combining the enzymes and the open hardware devices created by the community, and others created by people in Argentina, other created by people here, like colorimeters, thermocyclers, and other machines.
That's fantastic to be able to kind of transfer the work that you've done in detecting SARS-CoV to detecting other diseases. So it's great to hear that those materials have been useful. I know that we were in a conversation with Jenny earlier on, and she was saying that we should look into kind of scaling down a little bit and essentially have this small subset of these are the most important and well-used enzymes, because I know there's always been a lot of requests for like PFU or BST-LF and HIV-RT. So maybe we make a more curated kind of, these are the ready-to-go enzymes to provide out. But yeah, definitely keeping in touch with, like, as you were saying, for now, we've been trying to set up the hub in Argentina. So it's first getting those collections over to them, to then make best use of and cut down on that kind of supply chain issue and things like that.
I agree. I think that there are many enzymes and components in the collection that sometimes could be helpful, and I agree that having access to, for example, MMLV and HIV RTs, and then BST and OpenVent and PFU, and also the OpenTag now, is very useful because it's what most of the people will require, and that's okay. But then, for example, in other projects, we for example, we have a problem with, we wanted to express a lactonase for another project related to biophysics research, and we couldn't get it to work. And what Ariel did was to use some of the tags in the collection of the Reclone collection and make it work. And that was thanks to the existence of those tags, so we could rapidly make combinatorial assemblies and then try some of them. Some did work, some other didn't work. So that was very useful to have access to this sort of tags and techniques, especially for the lab, as Ariel mentioned at the beginning, for a lab that has not specialized on protein production. We collaborate with Cesar, but our lab doesn't work on protein production. So having access to this Reclone collection was super useful. Ariel can tell you more about what tags did he used for this.
Yeah, I was just going to say it would be lovely to hear more about this work as well.
For lactonase or another one?
Yes.
Oh, okay. Well, I only use the available collection because, as Fernán says, I'm very all my work, my PhD work is with modular cloning. So when I saw that this collection available, not for only clinical purpose, I see a lot of options for tags. So I just use it, and we assemble 4 or 5 options, and we tested along, and they at least one worked well at the first. So for me, it was very useful. Indeed, in my actual work that I'm doing synthetic circuits to modulate the trade-off between growth and stress implant, I already using some part of that that kit. So as Fernán said, this kit not only allows to use it in a context for clinical purpose, but is universal, and the modular composition of it as even allow that even let me use it in a context completely different.
I'm glad to hear that they've actually been really helpful. It's one of the things that we've been quite interested in, is just to reach out to people who have used the kits, used the collections, and just find out a bit more, essentially make use stories, use cases to say, look,
Okay.
you know, Ariel's been working with these 4 tags for the lactonase. It'll be great to hear, you know, the benefits of this, so that more people can see what the different possibilities are. And I think that'll couple really nicely with the education resources that, I mean, the training that you guys have done, and what Fernán you're doing with the Argentinian groups. It'll be nice to see and share those resources and kind of see how we can best help support people when using these open collections. Sorry.
By mail, I can provide a lot of examples where I have used that kit. So let me know, and I can write something.
Yeah, definitely. I'll keep in contact and find out more. And if you want, just post directly onto the forum as well. I'm sure a lot of people will be interested. I think one of our plans is to set up like a kind of informal holding station section on the category. Holding category on the forum where people can post up protocols, where people can troubleshoot, and eventually we'll move all that onto the protocols.io, like you shared earlier as well. I'd love to dive a bit more into the educational resources, but I've just seen in chat that Adrian, you had a question. I may have missed that earlier, sorry. But it was if Ariel Fernán, if you guys had any plans to extend to other pathogens, but you've also mentioned it earlier with the waterborne diseases. So yeah, sorry, I just missed your chat entirely. So yeah, are there any other questions in the room for Ariel or Fernán about the current research? And if not, then I'd be happy. Well, first of all, thank you, Ariel, for presenting again about your work. Big round of applause for you.
Thank you for inviting me.
No, it was good to hear. It's nice to see from people all over the world the different work that's been done. As I said, it's good to gather those kind of case studies. So now I guess I want to open the floor to anyone else in the room who want to update us about what else they're going on, what else they've been doing recently that they'd like to share to the community. No worries if there are no major plans. It's always just nice to hear. Sorry, Joseph. Feel free to share, Joseph.
Okay, I want to share that because the work is going, but it is very slow with the PFU enzyme. But we found that it could be, we compared it to the tag polymerase, and it could very easily amplify 2.5 kilobases with ease. I think we could step it up and reach some 5 kilobases of PFU, and I think then we should do some screening. We don't use sequencing, we use some, make sure that the sequence is right after amplification. But I think the PFU, the SSO7D part could amplify very large sequences. I think that could be applied in, and even also trying to produce a whole enzyme, whole plasmid, some small plasmids with ease. So this is what I can share for now, and I hope with further results I can also share with you also.
Yeah, definitely. That's fantastic, Joseph. I mean, 2.5 kb is quite a long stretch, and especially if you can increase the processivity. Adrian.
Yeah, so I was working on a hardware stuff based on Sebastian's Cochaba stuff. It's a transilluminator. I'm not sure if you guys are interested in that kind of stuff, but it's like a homemade, and it's it's working. Just give me a second.
As a sneak preview whilst Adrian's getting stuff, Adrian will also be our speaker for next month. So just a heads up.
Yeah, sorry. So this is the transilluminator, and it's a dedicated PCB board with some LEDs. And actually, one second, to get it off.
No worries. Era, do you mind if you stop sharing screen so we can see more of what Adrian is also sharing? That's okay.
Sorry. So basically, how do I do this so you can see it? Okay. So the board looks like this, and the top looks like this. And if you put the top on, okay, now you cannot see anything, but on the top there's you can put your cell phone in here. You will get the light in here, and actually this goes perfectly over the these holes in here. They will go over the cables. So actually, the interesting part about it, because I was thinking maybe I can film directly when the gel goes, and we might be able to use an algorithm to use the motion instead of just the plain the plain picture and correct the sensitivity. So if I do that, I hope to get a sensitivity much better, much clearer bands than even the commercial transilluminators that are right now. I think it's like, I don't know, maybe 20-30 bucks to make, and you can do it at home, and it's yeah, it's a nice thing in case any one of you is interested. Yeah, the PCB board, yeah, it's about $5 for 5 of them, so that's good.
Oh, that's amazing. That's very cheap to make your own transilluminator. I mean, I remember I was trying to cut out a cardboard box just to cover things, like over things. So it's definitely a scale up from that, but also very affordable.
Yeah, so initially I made one box, like one complete box, and then I realized, oh, just a second, what if I want to cut the gel? And then I have to make it to open, so and still leave the transilluminator inside, so we can still what's going on. The work actually is based on Sebastian Kochaba's tear down of actually a commercial transilluminator, and I stole from there the LED's frequency, the LED types, and the filter. There's another yellow filter that's just the plexiglass. But yeah, this is much better, and I'll publish it on in open source. But if anybody's interested, I can send you materials.
No, that definitely sounds really fascinating. So, like anyone who shared already here today, feel free to post any new updates you have on the forum. I also believe that the GOSH forum, the Gathering for Open Source Hardware, will be very interested in what you're doing here too, Adrian. So we'll make sure to talk with Bria and cross post that over there as well, if you're not already on there.
Okay.
Yeah, that's some nice updates. And as I said, this is a sneak preview because Adrian is our guest speaker for June. So it's a bit of a show for what's coming up. Cool. Anyone else in the room want to share what they've been doing recently? Or should I give you back 6 minutes of your hour? Okay. So seems like that's it for now. So I want to thank Ariel again for presenting to us today, and for all you guys for joining us and sharing your updates as well. Without much else then, wish you a very good morning, evening, good night, wherever you are, and I hope to hear from you guys again soon. Take care now, guys. Bye.
Thank you all.
Bye.
Bye.